genetic profiling. You can even call it as a DNA fingerprinting. Okay, and this includes gel electrophoresis, right? So as what I have explained you what is gel electrophoresis already, right? So if you get a blood sample, now example, if you go into a crime scene and you see some blood spots has been detected from that murder area, so we take the blood sample, we extract the DNA, from the blood sample, and we will cut the DNA into fragments, right? By restriction endonuclease enzymes. So when I say DNA fragments, as I told you earlier that restriction endonuclease enzyme cleaves it closer to the VNTR regions. So we know what is VNTR. VNTR is variable number tandem repeats, right? And when they ask you what is variable number tandem repeats, a region of DNA which contains variable numbers of repeated DNA sequences. So that I have already explained you in the previous class, right? So we have the restriction endonuclease enzyme going to cut the DNA closer to the VNTR regions, right? Now the DNA is ready for the gel electrophoresis process, right? So what we are going to do is we are going to put these DNA fragments into the wells. Now think this is your gel electrophoresis tank. Right, this is your gel electrophoresis tank, and we are going to put these DNA fragments into the wells that is present at the cathode area, because cathode is negative, and we know that DNA is a negatively charged particle, right? So we are going to put to the wells here in the cathode area, and current is applied. Okay, so as we have already studied that in gel electrophoresis tank we have agarose gel. If we are using DNA, it has to be agarose gel. If you are using proteins, it has to be polyacrylamide gel, right? So now we have placed the DNA onto the wells of the gel electrophoresis tank, and the current is applied, right? So the DNA fragment starts to travel towards the anode. Now imagine this is your cathode area, this is the anode area. So now the DNA fragments used to travel, right? So we also have learned a fact that if it is shorter DNA fragments, it will come very fast and further, like it will come very much far from the cathode. It will come more towards the anode area, right? But if it's a very large DNA fragment, it will move slowly and you would be able to see a band over here, right? But as I told you that now in the gel electrophoresis tank, even though the fragments are moving, right? The fragments are moving, but you can't see the visible bands in the gel electrophoresis tank. It is impossible. You can't see the black color bands as what we saw in the like this bands. You can't see it here, right? So that's what we are going to do is we are going to transfer these bands onto a absorbent paper, right? So we are going to place the absorbent paper on top of the gel tank, or on top of the agarose gel. We are going to place the absorbent paper on top of the agarose gel. So when you place it like that, the bands that the DNA bands, I mean the DNA fragments which has moved, will be transferred onto the absorbent paper. Then what we are going to do next is we are going to heat the absorbent paper. Now, why we are going to heat the absorbent paper is we want to make that DNA strand, the two DNA strands to separate from each other. Like this absorbent paper, we are going to heat it to separate or to make the two DNA strands to separate. Right? Now, why we are making it to separate, you will understand next. Now, we are going to make it single-stranded. In other terms, we can say we are going to make the double-stranded DNA into single-stranded DNA. So, that's why we are heating the absorbent paper so that double-stranded DNA fragments will be unwinded and it becomes single-stranded. Okay? After that, I am going to add probes. Right? So, when I say probes, it is short sequence of single-stranded DNA. I am going to add probes, meaning short sequence of single-stranded DNA, right? So, when I add the probes, now as we know that the DNA fragments are also single-stranded now. Now, probes also single-stranded, right? And you have to know the fact that probes have base sequences complementary to the VNTR regions. So, as you all know that now normally DNA fragments are now VNTR regions, right? Because the restriction enzyme has cut into VNTR regions. So, the probes also have base sequences complementary to the VNTR regions. And also you have to know that probe also contain a radioactive phosphorus isotope. Radioactive phosphorus isotope. Okay. So then when I add the probes which is having a complementary base pairing with the DNA fragment, it will form. It will form a complementary base pairing with the DNA fragments, right? Then next what we are going to do, now that's already done. Now the probes we have added and the probes have formed complementary base pairing with the DNA fragments. But still you can't see the bands. Then what we are going to do is we are going to put the absorbent paper, place it on the X-ray film. We are going to place it on the X. Now this is the absorbent paper. The X-ray film is dark. So you are going to put the absorbent paper on top of the X-ray film. So when you put on the X-ray film, what happens is, now as I told you that probes have a radioactive isotope, it will try to emit. The probe which has formed complementary base pairing with the DNA fragment, those probes will try to emit radiation. So when it emits radiation, the X-ray film will get dark colors. So example, just think you have a DNA fragment which has formed a complementary base pairing with a probe in this area, right? So when that probe is emitting the radiation, that radiation is making a dark film on the X-ray film, a dark band on the X-ray film. So that happens to all the fragments. That is, now example, you have a fragment here, another fragment here, another one here, another one here. So only those probes which has formed complementary base pairing with the DNA fragment will emit the radiation, and that radiation will form a dark color band on the X-ray film. After that, you see the X-ray film where you will be able to see the bands like this. Right? And just think that now the bands I am showing you here, right? This is the DNA fragment bands are different from every person. Now example, the DNA bands that is shown here will be different from my one, from Shahania's one, from Situ's one. So each and every person, this band pattern differs. So that's why we say the VNTR regions are different for each person, but they are identical for the identical twins, right? So this is how the genetic profiling, or you can even use the word DNA fingerprinting process happens. So this is how you actually view the bands. You don't view the band in the gel electrophoresis tank, right? You actually view the band in the X-ray film. So this is your X-ray film, right? Now I will show you another picture, this one. Okay, you can see here, using DNA profiling in paternity testing, right? So that means now a child is there, so they want to make sure if that child, now just tell there is mother and there is father A and father B, right? Two fathers are there. So this child, whether it belongs to father A or father B, they are not sure, right? So now maybe, like, normally these cases normally happen, right? So if they don't know, if the child really wants to know if he belongs to father A or B, what they have to do is they have to take the mother's DNA pattern, the father's, both the father's DNA pattern and the child's one, right? Otherwise, you can just think, now if the child thinks his father is father A, then only you can take the DNA pattern of the father A and check if the band pattern matches with his father. So this is what you see now. This is the X-ray film with the DNA band shown to you, right? Now here they have said control DNA. Control DNA means just we have taken a DNA from someone. We have just taken it from someone, right? Then we took that particular child's mother's DNA band pattern and that child, and this is the possible father. Possible father means we are going to see if this father belongs to him, right? So what you have to know is this particular child, he has to share his DNA band patterns of mother and father. It's not that he's only going to have the DNA pattern of the father. If that is the case, then they both are identical, right? So that's wrong. He actually has to sorry, he actually has to share the DNA band pattern of his mother and father. So first, if you just observe here, this is similar to the father but not to the mother. So that's fine. That means one is there. Now second one, in the second he doesn't, the father has but he doesn't have, no problem. You go for the third one. Third one, of course, matches with the father. Then go to the fourth one. Fourth one matches with the father. Go to the fifth one. Fifth one matches with the mother. Yes. Then go again to the sixth one. It's matching with the father. Seventh one matching with mother. Eighth one matching with father. Ninth one matching with mother. 10th one father. 11th one father. So now we can 100% prove that this particular child belongs to this mother and this father. So this is his real mother and father because his DNA pattern bands is sharing both. Okay. Now if you see, look at this one. This is also an interesting one. Using DNA profiling in crime scene analysis. So you can see here DNA fingerprint of a blood stain found at the scene of a crime compared with the fingerprints from 7 suspects. Now here they have shown you here the blood stain at the scene of crime. So that means they have collected a blood sample from that murder area. So that is this one, right? And they have suspect of 7 people. So that's why they have put 1, 2, 3, 4, 5, 6, 7. So 7 people, they have suspects, right? Now you see, if this DNA band pattern matches with one of the suspects, that means he is the murderer. So then you see now here, when you just take the person 1 and you try to see if they both are same, the pattern, the band pattern of person 1 is not same as the blood that they got at the crime place, right? So not the murder. So then second one you see, second person also not the same. Third one you see, it is same. Here you have same, the fourth one, fifth one, sixth one, exactly the same as the crime where they got the blood at the crime scene. So which means the person 3 is the murderer. And if you still want to go on to the next suspects, you can check the fourth one different, fifth one different, sixth one different, seventh one different. So only one suspect's blood DNA pattern matches with the blood DNA pattern that they got at the scene of the crime. So which means he is the murderer, person 3. Right? Now, are you clear, children, of both what I explained?
Yeah, miss.